Buenos días, vamos a comenzar esta primera sesión de esta segunda parte del programa de este año. Está conectado ya nuestros colegas también del 12 de octubre. Si quieren hacer un comentario. Hola, ¿qué tal? Bueno,
buenos días.
Qué bien, ahora se oye perfectamente. Buenos días. Decía que para nosotros, for us, it's a great privilege getting again with us Professor Desmond Sheridan, that is Emeritus Professor of Cardiology in the National Heart and Lung Institute in UK. And he, for us, as I said, is really a privilege getting here in a new lecture about some issue related with the cardiovascular system, the evolution and the development of the cardiovascular system. And today the lecture is again related with this issue that is titled Blood and Heart, Coevolved Intimate Partners. And for sure, Desmond will give us some insights on the evolution and how we evolve the cardiovascular system. So Desmond is sometimes here in Santiago. He travels a lot, but he visits our city and our university at least 2 times per year. For us, it's a great privilege to get in here today in this session again. And now in this joint session, I was talking with Desmond. This is a joint session with the Cardio 12 de Octubre hospital, that is a big university hospital, included also in the cyber network, research network in Spain. Our colleagues are attending from the 12 de Octubre hospital. So Desmond, thank you again for coming here and for being so glad to do so kind to give us this lecture about this, how to evolve the blood, the heart, and at the end, the cardiovascular system. Thank you for coming.
Jose, Professor Gonzalez, ladies and gentlemen, thank you for inviting me to come back to your beautiful city to speak to you again about my interests in the heart. In my previous talks, I talked about the cultural origins of the word heart and how the heart evolved following the onset of photosynthesis. But today I want to talk to you about one of its close partners, and I will show you how the structure and physiology of blood have evolved to match those of the heart and blood vessels, and why this was essential for the efficiency and durability of the circulation. Blood has many functions, most notably as a transport medium for nutrients, waste products, hormones, signaling molecules, but also as mobile components of the immune system, coagulation, and thermoregulation. But acting as a mechanism for oxygen transport is perhaps its most immediately pressing function of blood. Oxygen enables aerobic metabolism, which provides about 18 times more energy than anaerobic metabolism, and thereby facilitated the evolution of large and complex animals. This slide summarizes the emergence and rise of atmospheric oxygen following the onset of oxygenic photosynthesis 2.5 billion years ago. And I've shown this slide here before. The red and green lines represent the upper and lower estimates of oxygen atmospheric levels, and this rise in atmospheric oxygen led to a great diversification of living organisms known as the Cambrian explosion around 550 million years ago. And from around 500 million years ago, whole body circulations which could carry oxygen to all parts of the body evolved. This was made possible by the evolution of circulation and oxygen carriers, of which 3 have evolved. Hemocyanins evolved around 740 million years ago, and they're present in arthropods and mollusks. Their oxygen binding sites contain 2 copper ions, which are held within a histidine ring. Hemocyanins are thought to have evolved from the oxidizing enzyme tyrosinase. In humans, tyrosinase oxidizes the amino acid tyrosine to form melanin. Hemoerythrins are also oxygen carriers present in marine worms and brachiopod shellfish. They are present in the fossil record from around 550 million years ago. Despite the name, they do not contain a heme group, but do contain two iron ions held within a histidine ring structure at the oxygen binding site. Hemoerythrins have greater affinity for oxygen than hemoglobin, suggesting that they may have had a role as oxygen storage mechanisms, for example, in burrowing animals living in oxygen-poor conditions underground. Hemoglobins are truly ancient proteins, and they're found in all kingdoms of organisms, including plants, animals, fungi, protists, and bacteria. They serve as oxygen carriers in almost all vertebrates and in some invertebrates. In some plants and bacteria, hemoglobin has evolved as an oxygen storage mechanism, while in others it acts as an oxygen sensor, and yet in others as an oxygen scavenger. In mammals like humans, it's always present within erythrocytes, while in many invertebrates it's present extracellularly. The complete structure of hemoglobin varies widely between species. However, they all contain a heme group with an iron ion at its center, surrounded by a globin protein, which has characteristic and well-preserved features. So you can see the iron ion is held at the center of histidine rings, and the globin molecule in which it's contained is very characteristic and well-preserved in nature, consisting of 6 to 8 of these these spiral structures, which are common in protein structures, held together with a pocket in which the heme group sits. Oxygen is capable of 6 coordinate bonds, of which 4 are linked to nitrogen atoms in the histidine ring, and the 5th is linked to a very specific and unchanging amino acid in the globin chain, while the 6th binds to oxygen. Globins are proteins of 17 kilodaltons molecular weight, and they have 6 to 8 of these alpha helices structures in a layered structure with a pocket which contains the heme group. As I say, the structure of globins is the most conserved of all known proteins and indicates a truly ancient heritage, most likely formed in a bacteria that lived around 3.5 billion years ago. Globins are present in a wide variety of polymeric forms, so although the globin chain is very constant, it's also present as polymers. In vertebrates, globins are present as single proteins in, for example, myoglobin in skeletal muscle, but as tetramers, meaning 4 linked chains in hemoglobin. However, in mollusks and in many worms, they are present as single proteins or monomers. Clams have dimeric hemoglobin, meaning 2 globin chains, while in earthworms, hemoglobin is present as dodecamers held together in groups of 12, and that literally is 144 globin chains all bound together in this absolutely gigantic molecule. Thus, hemoglobin is found in many forms in nature, and these reflect the many specific roles it plays in different species. In this talk, I hope to show you that the molecular configuration present in humans has evolved to exquisitely match the needs of the heart and circulation. In all animals, oxygen transport is the primary function of hemoglobin, and together with the heart is key to the availability of energy and oxygen consumption and energy. Not surprisingly, energy production and therefore oxygen consumption is related to body size, as illustrated in this study of mammals and birds, with a wide range of body weights. This holds true for oxygen consumption at rest and also at maximal oxygen consumption, clearly related to body weight, and this is on a log scale, so this is a very big range of body weights. Aerobic range, on the other hand, refers to VO2 max, or maximal oxygen consumption, related to resting oxygen consumption. In other words, by how many times can an animal increase its resting oxygen consumption or its basal metabolic rate? For example, trained footballers can increase their basic metabolic rate about 20 times, meaning they have an aerobic range of 20 compared to around 12 in sedentary persons. In a study of several species of mammals and birds, Bishop examined how aerobic range is related to body weight. And as you can see here, the data are widely scattered with no significant relationship to body weight. But this is not unexpected, since oxygen consumption is more likely to be related to lifestyle than size. For example, a small bird with the demands of flying will expend more energy for its weight than a large, slow-moving mammal. However, if we look at aerobic range in relation to body weight while controlling for heart rate and hemoglobin concentration, then the data do show a significant relationship to body weight, as shown here. And this tells us that the main determinants of aerobic range are heart size and hemoglobin concentration. In the study I just showed you, around 85% of the variability in aerobic range was attributable to heart size and 15% to hemoglobin level. And a contribution of just 15% might suggest that the role of hemoglobin is relatively minor. However, in the study I just showed you, it was limited to birds and mammals, and in reality, hemoglobin concentration doesn't vary much amongst all terrestrial animals. If instead we look at a wider range of aquatic and land-based animals, we can see a much greater range of hemoglobin concentrations from just 4 grams per deciliter in hagfish and sharks to 13 to 15 grams per deciliter in humans and Weddell seals. And so as animals evolved larger and more complex bodies, bigger and more powerful hearts made this possible, but so too did increasing oxygen carrying capacity due to higher hemoglobin concentrations. Interestingly, Weddell seals and killer whales, as illustrated here at the bottom of this slide, have evolved an additional mechanism by retaining a store of oxygenated hemoglobin in the spleen, which they can release when the demands of oxygen are increased during diving. So, as I mentioned, the globin proteins present in hemoglobin are extremely constant in nature. However, the overall structure of hemoglobin varies greatly among species because of the different polymeric forms. As I mentioned above, vertebrate hemoglobin is tetrameric, consisting of 4 globin chains. But hemoglobin is also found as single, double, and multiple polymers. As an example, the very large 144 globin chains present in earthworms. Such large polymers of hemoglobin can carry more oxygen per molecule, and they can do so extracellularly because they're so large they don't leak from the circulation. However, there is an important trade-off against viscosity. And this is because viscosity increases steeply with both concentration and molecular weight. This has an important effect on blood perfusion because viscosity is positively related to vascular resistance. And blood viscosity increases, so too will vascular resistance, and in turn, the work required of the heart to pump it. This becomes a limiting factor as hemoglobin concentration rises, and it's for this reason that each molecular weight of hemoglobin, there is an optimal concentration for oxygen transport. So as shown here, with increasing concentrations of hemoglobin, oxygen transport can increase. But this also reaches an optimal value because as the concentration rises further, viscosity increases and this limits perfusion. And in reality, the optimal concentration is much better for lower molecular weight hemoglobins than very large molecules. And 68 kilodaltons represents the tetrameric form found in mammals, including humans, which has an optimal carrying capacity at around 15 grams per deciliter of hemoglobin. as compared, for example, large molecules where the optimal carrying capacity is at just 5 grams per deciliter. Consequently, the evolution of animals with high energy needs requiring greater oxygen transport was facilitated by having higher hemoglobin concentrations, and this in turn was made possible by having hemoglobins with low and relatively low molecular weight and viscosity. Of the many molecular forms present in nature, tetrameric hemoglobin is ideally suited for mammals with high energy needs by optimizing transport with only modest increases in viscosity and vascular resistance. And so we can see that the precise form of hemoglobin present in humans has evolved to best suit the needs of our hearts and circulations. A key property of hemoglobin is that its oxygen dissociation curve. That is, how tightly it binds and releases oxygen. Tetrameric hemoglobin is capable of a wide range of oxygen dissociation curves. In tetrameric hemoglobin, the 4 chains are closely interlinked and bind together, as in this example of adult hemoglobin, alpha-2, beta-2 hemoglobin, in which the 4 chains are closely interlinked and bound together. But importantly, hemoglobin is a highly allosteric molecule, meaning that changes in the conformation of one element have effects on other parts of the molecule. It means that the strength of binding between the 4 chains affects oxygen affinity inversely. The stronger the binding between the globin chains, the weaker is oxygen affinity, and vice versa. And small changes in the amino acid sequence of the globin chains affects mutual binding and therefore oxygen affinity. And it's for this reason that hemoglobin has evolved with a wide range of oxygen affinities to suit different circulatory conditions. In humans, hemoglobin synthesis begins in the yolk sac, producing epsilon and zeta hemoglobin chains for the first few weeks of life. Later, the liver, spleen, and bone marrow take over this role. The globin chains produced at each stage have only minor differences in amino acid sequence. However, they result in marked differences in oxygen dissociation curves. So adult alpha-2 beta-2 hemoglobin, as shown here in the bottom slide, the rightmost curve, is 50% saturated at a partial pressure of oxygen of 25 millimeters of mercury, whereas embryonic hemoglobin, zeta-2 epsilon 2 hemoglobin, is 50% saturated with a partial pressure of just 5 millimeters of mercury. And these, of course, are ideally suited for the hypoxic conditions faced by the early embryo, as well as the more oxygen-rich conditions of the adult circulation. Again, we can see how the evolution of hemoglobin has evolved in such a way that it perfectly matches the situation in the circulation. As you well know, erythrocytes are biconcave discs, and this offers many advantages for the circulation. Due to the distribution of mass at the periphery, biconcave discs are more stable in the circulation than if they were spheres. And this is because it takes less energy to rotate a sphere than a biconcave disc. And for this reason, biconcave discs are less prone to rotation in the circulation, and this minimizes turbulent flow, which protects the circulation from platelet activation and diseases such as atheroma. The normal hemoglobin concentration in whole blood is around 13 to 15 grams per deciliter. However, hemoglobin is packed into erythrocytes at a much higher concentration, so the MCHC, the mean corpuscular hemoglobin concentration, is around 35 grams per deciliter. Recently, it has been shown that this high MCHC greatly reduces the diffusibility of gases within red blood cells. For example, carbon dioxide diffusion in water is 20 times greater than within red blood cells, and this limits gas diffusion between hemoglobin and tissues. And for this reason, diffusion distance between hemoglobin and tissues is of great importance. And the biconcave shape of red blood cells is a major advantage in this regard because it reduces diffusion distance between hemoglobin and the cell membrane. The biconcave shape also facilitates folding of erythrocytes, as illustrated here. On the lower right, we have an electron micrograph, which I've simply redrawn here for illustration purposes. This reduces resistance to flow in small vessels and thereby pressure needed to maintain the circulation. It also minimizes the diffusion distance between the surface of the erythrocyte and the capillary cell membrane. And this also assists gas diffusion. Viscosity of blood is a major factor in resistance to flow. The viscosity of blood is about 4 times that of water and is affected by several factors such as higher hemoglobin concentrations in polycythemia and also at cold temperatures. The viscosity of blood is also affected by flow rate because when static or flowing slowly, red blood cells tend to adhere to one another, forming stacks or rouleaux, which also increases viscosity. But perhaps one of the most remarkable properties of blood is that its viscosity is reduced when flowing in small vessels, such as arterioles and capillaries. This is because of the folding property of erythrocytes, shown here, which reduces resistance to flow, and because red blood cells tend to occupy the center of the lumen surrounded by plasma. This is well known as the Farhas-Lindqvist effect, and it means that when blood is flowing through small vessels, the size of arterioles and capillaries, it has a much lower viscosity than in larger ones. This in turn has a major effect on the heart and circulation because small vessels vastly outnumber large ones in the circulation, as illustrated here. This is based on the circulation of the dog. The blue bars indicate the number of vessels at each size, and it scans the circulation from the aorta right across to the vena cava. And as you can see, the numbers of small vessels in the range less than 200 microns vastly outnumbers the others. And this is on a log scale on the left-hand side. So we're talking about hundreds of millions of vessels in this size range. This also means that the distance blood travels in small vessels is much greater than in large ones. And this is illustrated here. So the collective length of blood vessels, here shown in blue, is much, much greater when traveling through in small vessels than in large ones. And as a result, the viscosity of blood is lower during most of its transit through the circulation, again known as the Farhas-Lindqvist effect. Again, showing the effect on viscosity. Vascular resistance is a major determinant of cardiac work, and most resistance occurs at the level of small vessels, the size of arterioles or less. And therefore, this reduction in blood viscosity in small vessels also reduces systemic vascular resistance and cardiac work. It has been estimated that without the Farhas-Lindqvist effect, the heart would need to generate a pressure of around 200 millimeters of mercury just to maintain the circulation. And so we can see that the way red blood cells have evolved is exquisitely matched to the needs of the heart and circulation in ways that greatly support its function. A crucial element of oxygen uptake and delivery to tissues is gas diffusion. And this needs to be optimal, and that the diffusion distance between hemoglobin in erythrocytes and tissues needs to be minimized. This is made possible by close apposition between the surface of red blood cells and the endothelial cells, as I showed earlier here. As you can see here, the ability of erythrocytes to fold ensures that the membranes lie close to the proximity to the endothelial cell membranes, which greatly enhances oxygen and carbon dioxide diffusion. This also implies that the size of erythrocytes and capillaries should be closely matched to minimize diffusion distance. However, as this slide shows, which I have redrawn from the work of George Gulliver, who published in 1867 this remarkable slide, which is well known amongst hematologists. He was an anatomist based in London, and he went off to the London Zoo and got blood samples from all the animals there and studied their erythrocytes. And as you can see, there's a huge variation in the size of erythrocytes. For example, salamanders have erythrocytes that are 10 times the size of mammals. However, irrespective of the size of erythrocytes, in many species, they remain closely related to capillary diameter over a range of shown here. So among salamanders, lizards, frogs, and mammals, there's a close relationship between erythrocyte size and capillary diameter. An important aspect of the circulation that as the diameter of capillaries is reduced, their density in tissues increases. So mammals have nearly 800 capillaries per cubic millimeter compared to just around 100 or less in mammals and in frogs and lizards and salamanders. And therefore animals with smaller erythrocytes have smaller capillaries and greater tissue vascularity. This also means that they have greater surface area for gas exchange and shorter diffusion distances. So again, we see in these groups of animals, capillary diameter and oxygen diffusion distance are related. So smaller capillaries, smaller erythrocytes have a shorter diffusion distance. Again, all enhancing high oxygen uptake and delivery needed by energy-demanding animals. Thus, evolution of erythrocytes seems to have occurred alongside that of the heart and circulation, so that their size matches perfectly that of capillaries of the circulation to ensure maximum uptake and delivery. And this, of course, is perfectly suited to the work required of the heart to pump as an oxygen pump. As I mentioned earlier, erythrocyte size varies greatly among species. And as you can see here, so too does nuclear size. What then does this mean? The human genome consists of around 3 billion base pairs, but it has been known since the 1950s that genome size varies greatly among species, with variations of up to 200,000-fold being reported. For example, the genome of a primitive lungfish has 43 billion base pairs, and salamander genome has 30 billion. That's 10 times larger than mammals or humans. Not surprisingly, genome size also correlates positively with erythrocyte volume. If you have a bigger nucleus, you need a bigger cell to accommodate it, as shown here for reptiles, birds, and amphibians. And although mature mammalian erythrocytes have no nucleus, their size also correlates with genome size, reflecting the fact that they were once nucleated. What is perhaps less expected is that genome size also varies inversely with heart size among species, as clearly shown here in this work by Vinogradov and Anatskaya. Heart weight to body weight ratio is clearly inversely related to genome size among birds, reptiles, mammals, and amphibians, so that birds and mammals have a much smaller genome size and a larger heart weight to body weight ratio. Increased use of oxygen was a crucial factor in the evolution of animals with high energy needs. As we saw earlier, erythrocyte size plays a key role in the efficiency of oxygen uptake and delivery. And this led to the intriguing hypothesis that perhaps reduction in erythrocyte size and capillary diameter may have been important evolutionary steps in achieving the higher oxygen uptake and delivery needed for high energy using animals such as mammals and ourselves. To study this, Hutton, Locker, and Farmer used fossilized bones to study the microvasculature of extinct ancestors of mammals and dinosaurs. Bones, like other tissues, need a blood supply, which is provided by a network of Haversian canals through which even the smallest blood vessels pass. Among animals living today, who are living now, the size of these canals corresponds closely with the size of capillaries and erythrocytes, as illustrated here for amphibians and mammals. Amphibians with larger erythrocytes and larger Haversian canals, larger capillaries compared with mammals. Studies of the canal structure and size in fossil bones from several extinct forerunners of modern mammals and birds does indicate a gradual diminution in the size of these Haversian canals over the past 300 million years. And this appears to have been the case for synapsids, shown on the left here, representing early mammals, and ceratopsids, representing early reptiles. And assuming that the canal size corresponds to erythrocyte size and capillary diameter, as it does in animals living today, it implies that erythrocytes have also become smaller in size over the past 300 million years. It's illustrated in this slide from Hutton, Locker, and Farmer. In the slide, it illustrates the evolution of mammals, of early mammals and on the left, and on early reptiles on the right, over the past 300 million years. And the color code is such that red indicates capillaries and erythrocytes of smaller size, with green being the larger size. And you can see a tendency towards a reduction in capillary and erythrocyte size over the past 300 million years. So in conclusion, ladies and gentlemen, increasing energy production has always been a strategic element in the evolution of larger and more active animals. The availability of oxygen was a key aspect of this. Hearts evolved to distribute oxygen to tissues and body plans of increasing size. And as it did so, it co-evolved with several partners, which greatly improve its efficiency and protect it from harm, notably the blood vessels, the autonomic nervous system, and the multiple sensors which provide information about its function. Blood has, of course, many functions, but most importantly, as an oxygen transport system. In this, it has evolved as an intimate partner of the heart and circulation. Hemoglobin is present in all life forms in multiple molecular configurations, suitable for the needs of each. In humans, the tetrameric molecular structure of hemoglobin is perfectly suited for transporting the high levels of oxygen needed, as well as minimizing vascular viscosity of blood. Erythrocytes provide the necessary packaging for low molecular weight hemoglobin, which prevents its loss from the circulation. Erythrocytes vary in size greatly among species, but in each case the size is ideally suited to the circulation. In mammals, the nucleus of erythrocytes is ejected during development, and in humans, their biconcave shape facilitates folding, which lowers viscosity of blood in small vessels and resistance to flow, thereby greatly mitigating the work required of the heart to pump. Thus, we can say the composition and structure of blood is exquisitely matched to those of the heart and circulation as a result of millions of years of intimate co-evolution. Thank you.
Thank you, Desmond, for this really exciting lecture on the how to evolve the heart, the blood, and the blood components for improving the energy efficiency in the body, in the humans and other animals. Any comment of questions for our colleagues from the 12 de octubre? I would like to put a first comment or question to you. According to how to evolve the blood and the heart, the relationship, it seems that the evolution of the heart and the blood in the human beings, if you have to decide in a near future or in a future how to more efficient evolve the blood or the blood components for improving not only the energy delivering efficiency, but also the avoiding, for example, chronic diseases, because the humans were really well adapted for fighting against the acute diseases like hemorrhagia, bleeding, or like fever or other acute conditions, but not to fight against the chronic conditions, because you use the same mechanism of in this case, not protection in the chronic condition. For example, in heart failure, we use neurohumoral activation that increases the rate of heart disease, and in this case, increasing the risk of death. How did you decide which type of changes do you decide to do in blood, in the heart, or in this concept of the blood, not only of the energy transported, but also an element that can be involved in chronic conditions?
It's a very interesting question to predict what evolution should do and will do in the future. And of course, I mean, we are clever as human beings, and as we work harder and harder and harder, we can begin to understand a little more and more about what evolution has done in the past. But it's another step to take on the role of deciding what evolution should do in the future. And I'm not sure that human intelligence is capable of such a task. However, what we can say is that in general terms, evolution is something that takes place over millions of years. I mean, hundreds of millions of years. Some of the biggest changes that affected human health have happened in hundreds of years. And we, as mammals, have evolved as hunter-gatherers. Our genes have evolved to allow us to lead as we emerged 100,000 years ago. Most of the chronic diseases that we see today are actually linked to the way we live and the lifestyles that we have. And I would say that these lifestyles vary greatly from the way we have evolved. So it's not so much that our evolution has got wrong, but the way we live has diverged from the way we have evolved to live as our ancestors did. So our diets, our exercise, our use of tobacco, all of these things. And for evolution to catch up with those challenges would be very difficult because it's something that takes place over, as I say, hundreds of millions of years. And so it needs a very long-term view of that. It would be better to ask ourselves, how can we live lives to suit the genes that we have now? And that would be much more successful. But it's a fair question. How could the circulation be more efficient? It's difficult because it's almost perfect. You know, everything that you interfere with is like it makes it worse. So when we tried to give inotropes to treat heart failure, it didn't work. When we offload the heart and give drugs that minimize its work, it actually does better. But what we're doing there is we're actually mimicking what the circulation actually does naturally anyway. So perhaps the best answer I can give is to learn more about how the heart and circulation work at a fundamental level and use that to guide us how we should live and treat ourselves. But yeah, and I haven't answered your question, but I don't think anyone can.
Yeah, because it is really an important issue, because when you suffer a chronic conditions, for example, heart failure, you use the mechanism of adaptation, in this case, like in the acute diseases. You induce activation of the sympathetic nervous system, the neurohumoral activation of rerunning. And not only these systems help us to maintain the life, but increasing the velocity to the death. So it's curious that the human beings, where they are suffering, for example, the same in cancer. When you suffer a cancer, the mechanism of adaptations to cancer not only protect against the cancer, but in some cases facilitate the metastasis in some issues. And this is really an interesting thing, because can be like a philosophic issue, because you are not able to reproduce a healthy species, and then nobody knows which that delete you from the possibility of reproduce the species with chronic conditions. In this case, we are able to put in actions really good mechanism of protection against the chronic conditions. And this could be, but in this case, the approach of heart failure. We are blocking the mechanism, not adaptation, but in this case, the deleterious mechanism. And in this case, it's the same in cancer. In chronic conditions, we are not good mechanism of protections. The mechanism that you develop in these conditions not only protect you, but also increase the rates of mortality. but it's really like a philosophic issue, but it's very interesting that should be that in near future or in the future we can be able to get a mechanism of protection against the chronic conditions. Juan, we have a comment from
Buenos días.
Yeah, Juan Delgado.
Hola.
Hola.
¿Nos oís bien? ¿Sí?
Perfecto, sí.
Bueno, pues nada, en primer lugar quería agradeceros aceptar este nuevo formato de sesiones. Realmente quiero dar las gracias personalmente a Iago por su implicación casi personal en que esto salga bien. En segundo lugar, aquí estáis viendo solo la mitad del servicio. Estamos en una situación de tránsito entre el área de hospitalización y el área de consultas y bueno, pues realmente la distancia casi es medio kilómetro y por tanto, bueno, pues aquí están los médicos más implicados en el área ambulatoria. Y en segundo lugar, lo que sí que me gustaría es agradecer al Professor Sheridan. Thank you for coming and thank you for your excellent talk. And my question is related to the oxygen consumption arrest because if I follow the stock, there are a significant relationship between oxygen consumption arrest and body weight. However, in other species, no human, there are no relationship between oxygen consumption arrest and body weight. It's more related with heart weight and hemoglobin concentrations. It's difficult for me to understand this disparity in human species and non-human species. Could you explain, please, more in deep this concept?
I think in the slide that I showed, there was a significant relationship between body weight and resting oxygen consumption over a wide range of animals. So I think that's fairly clear. And humans are similar, I think. So I'm not quite sure that I'm picking up the question correctly. Perhaps you mean aerobic range. Are you perhaps thinking of aerobic range? Was that? If we go back to the slide, I can perhaps.
And can you hear the response of Desmond? Do you mean the aerobic range? Is the aerobic range?
Relationship.
In this slide, it's possible to. There are a significant relationship between body weight and oxygen consumption and rest. However, in the next slide, please. Yes, there are no relationship between body weight and the aerobic range.
Yes.
And the follow slide, please.
Follow one with the next slide.
This one?
Yeah, it's good.
Yeah, it's okay.
So to explain,
the relation.
Perhaps I'll just explain it again. So resting oxygen and maximum oxygen consumption, that's straightforward. We understand that. Aerobic range is the number of times an animal can increase its basal metabolic rate. So it's not actually maximum oxygen consumption or resting oxygen consumption. It's the number of times you can increase your basic metabolic rate. So can you double it, triple it, quadruple it, or multiply it by 10 times or 20 times? So as I said, a trained footballer can increase his aerobic range by about 20 times, whereas a sedentary person will increase it about 12 times. And if we look at aerobic range in a wide range of animals, there's no relationship to body weight. However, if we control for hemoglobin concentration and heart weight, then there is a relationship. So what it's telling us is that it's not body weight that's related to that determines aerobic range. It's heart rate and hemoglobin concentration.
Okay.
Okay?
Okay, thank you.
Okay, pleasure.
So thank you, interesting. So Desmond, thank you. This is really interesting because it is a kind of talks. It's a little bit away from our daily practice, but it's really interesting because we have to think about, in this case, how to evolve the blood, the heart, the body, and how with this knowledge we can, including plan new researchers for improving not only the efficiency, the energy efficiency based on the erythrocytes, oxygen delivered, but also how to get a better adaptation against the chronic conditions, because this is the major issue in the human beings. Not only a good response to acute conditions, because we are well prepared for a good acute response to acute diseases, but we have some problems for adaptation in chronic conditions, which is the reason why the mechanism of adaptations in chronic conditions like cancer or heart failure are not protecting us, are promoting the evolution of the disease. And this is a big issue, and we have not to wait on the thousands of years of evolution. We have to learn about these facts and be able to design new research for getting new mechanisms of protection against the chronic conditions. Yeah?
One point that I would like to make, a general point, is that as cardiologists, we think of the heart. It consumes us, the heart. But what I'm very keen for us to understand is that it's actually part of a big system. So we accept the heart is part of the circulation and the blood vessels. I like to think too of blood as part of that same thing, that blood is not a passive liquid that the heart, it's the object of the heart pumping. It actually is part of the whole thing. It's an integral part of it. And increasingly, I'm sure we'll find that the body as a whole is integrated in ways that we don't yet fully understand and take into account. So, level of hemoglobin, obviously, but it affects how the circulation works, very much temperature, all of these things. And by having a broad view of this and thinking of blood as part of the circulation, for me, it's a broader view of physiology, but also it's aesthetically and philosophically more satisfactory to think of blood in this way. It's a more perfect whole approach to how evolution has evolved to bring us to the situation we are in, and how effective humans are and how successful they are.
No, so thank you. It's really interesting. I thank again for accepting our invitation to come again to our university hospital and to be here in Santiago with us. And this is almost your home. You can be here with us wherever you want. And all you need that we can help, please don't hesitate to contact us. And we remain in touch for future collaborations. Many thanks. Yeah.
¿Tienes que colgar? ¿Dónde colgar?
Ah, ya le he dado.
Ah,
ahora es sí.
sí. Ah, eso está.